Welcome to Bookfulness. This is our deep dive into Chapter 7 of Evolution. Now, in our last deep dive, we really focused entirely on natural selection, that directional force, the adaptation engine. Today, we're doing a complete pivot. We're looking at something equally fundamental, but, well, totally different. Random chance.
Yeah, exactly.
Okay.
Our mission today is really to get our heads around genetic drift. This is the idea that evolution can happen just by accident. It's a stochastic process, meaning random, where allele frequencies change, not because one's better than the other, but just because of luck.
Right, just sheer luck of the draw. And to really show how powerful this random force can be, I think we have to start with a pretty dramatic story. The northern elephant seal.
Oh, absolutely. Moringa angostirostris. I mean, these animals are incredible, and they basically survived by the skin of their teeth. Back in the 19th century, they were hunted almost to extinction for their blubber. The population crashed. We think, based on the evidence, the entire species might have been reduced to maybe just 20 or 30 individuals hiding out on one island off Mexico.
20 or 30. That's almost unbelievable. I mean, especially when you see them now. There's, over 170,000 of them. They've bounced back incredibly well in numbers. But the genetic scar is still there, right? The damage was done back then.
Absolutely. That population crash, that extreme reduction, is what we call a population bottleneck. And it was maybe a terrible thing for the seals, but a perfect illustration for us of genetic drift. When you squeeze a whole species through such a tiny number, like 20 individuals, most of the genetic variation, all those different alleles floating around, just gets lost. Poof, gone. Not because those alleles were bad, not selection.
Yeah.
Just because the few individuals who happened to survive didn't carry them. Pure chance.
So, okay, if 99% of the seals vanished, the 1% left were just a random sample. And whatever genes they happened to have, that was the entire toolkit for the next 170,000 seals.
That's exactly it. And when scientists looked, comparing the genes of modern seals to museum samples from before the crash, the difference is stark. Modern elephant seals have dramatically less genetic variation. That loss, purely from the random sampling during that bottleneck, is probably one of the clearest examples we have of drift's power. Chance wiped the slate clean, not selection.
Okay, so let's really unpack this. We keep saying random chance, luck. What does that actually mean, at the genetic level?
Right. So genetic drift, at its heart, is just changes in allele frequencies because of these chance events. It's totally separate from whether an allele is helpful or harmful, which is selection. When we say chance, we mean it's stochastic, there's randomness baked in. And importantly, this isn't some rare thing. Drift is always happening in every population all the time.
Always. So where's the randomness coming from day to day? It can't just be near extinction events. Is it just bad luck, like getting struck by lightning?
Well, yeah, that kind of thing can contribute. Survival accidents. An animal could be perfectly fit, but just be in the wrong place at the wrong time, gets caught in a flood, stepped on, whatever. That's one source. But there's a much more constant, fundamental source of randomness. The process of making babies itself. Meiosis.
The shuffling of genes.
Exactly. Think about it. If you're diploid, you have two copies of a gene, say big A and little a. When you make sperm or eggs, which allele gets passed on, big A, little a. It's basically a 50-50 coin flip for each gene each time. Now, in a huge population, millions of individuals making millions of gametes, all those coin flips tend to average out. 50% heads, 50% tails. The overall frequency of big A doesn't really change much.
The law of large numbers kind of protects it.
Precisely. But what if your population is tiny, say just 5 breeding pairs? And maybe one pair carries a brand new allele that's currently at 10% frequency. If that one pair just happens, by chance, to mostly pass on the other allele to their few offspring. Maybe they flip 4 tails in a row, metaphorically speaking.
Then bam, that new alleles frequency could plummet from 10% down to maybe 5% just because of reproductive bad luck in one generation.
Exactly. That's the core of it. The randomness of which allele gets into which gamete, combined with random who survives who doesn't events, means that in small populations, drift can cause huge swings in allele frequencies, even if there's absolutely no selection pressure favoring one allele over another.
Okay, so to really study drift without selection messing things up, you can't easily use wild populations. Selection's always there. So I gather scientists turn to computer simulations, where they can just switch selection off.
Yeah, computer models are crucial here. You set up an idealized population in the computer, maybe give it starting allele frequencies, and then you just let random sampling run the show generation after generation, no selection allowed. And doing this over and over with different parameters has really helped us nail down 5 key features of how drift behaves.
Okay, what's the first big takeaway?
Feature 1. Drift is unbiased. This is fundamental.
Mm.
In any single generation, an allele's frequency is just as likely to go up as it is to go down. There's no inherent directionality based on fitness, like with selection. It's what we call a random walk. Imagine plotting the frequency on a graph. It just sort of jitters up and down randomly over time.
So it doesn't want to go anywhere specific.
Nope. If you run that simulation 100 times, starting from the same frequency, you'll get 100 completely different paths. Some might go up, some down, some wiggle around the middle. But on average, across all those runs, the changes balance out. It's unbiased movement.
Okay, I like that. Selection is like gravity, always pulling one way towards better fitness. Drift is more like, I don't know, random gusts of wind, pushing you this way and that unpredictably.
That's a great way to put it. And that wind analogy leads right into the next absolutely critical point, feature 2. Drift is stronger in small populations. The effect size is inversely related to population size, n.
Right, we saw that with the elephant seals. Small n, big chaos.
Exactly. Think about flipping coins again. If you flip a coin 500,000 times, a huge population n 500,000, getting, say, 51% heads instead of 50% is barely a blip. The frequency is super stable. The random walk is just tiny little jitters.
The law of large numbers smooths it all out.
To flip that coin only 5 times, n5. Getting 3 heads instead of 2 or 3 tails instead of 2, that's a massive frequency shift, maybe 60% instead of 40%. When n is tiny, like 5 or 10 or even 50, each random birth or death, each random coin flip and meiosis can have a huge proportional impact. The fluctuations, the random walk, it becomes wild and extreme.
So population size acts like a damper. Big N dampens drift down to almost nothing. Small n lets it run wild.
Precisely. Population size is the key controller of drift strength. And these wild fluctuations in small populations lead inevitably to the third feature, which has huge long-term consequences.
Okay, what's that?
Feature 3. Drift causes loss of variation. This is a big one. Unlike some forms of selection, like heterozygote advantage, which can maintain different alleles, drift is purely destructive when it comes to variation within a population. Over enough time, that random walk we talked about is guaranteed to hit one of 2 endpoints. Either the allele's frequency randomly walks all the way up to 1.0, we call this fixation, or it randomly walks all the way down to 0.0 loss.
And once it hits either 1.0 or 0.0, it's stuck there.
It's stuck. If an allele is lost, frequency 0.0, it's gone from that population forever, unless a new mutation recreates it, which is rare. If it's fixed, frequency 1.0, then all other alternative alleles at that locus are gone. So the inevitable long-term outcome of drift in any single isolated population is the erosion of genetic diversity, the elimination of polymorphism.
And that's crucial because variation is the raw material for evolution, right? If you lose all the different alleles at a bunch of genes because of drift,
Then you don't have options. If the environment changes later and some lost allele would have been beneficial, well, too bad. The species might not be able to adapt because drift already randomly threw away the necessary tools.
creates a real vulnerability. And that ties into the next feature, which looks at multiple populations.
Yeah, feature 4. Drift causes divergence. Imagine you start with one big, happy, diverse population. Then you split it up, maybe by fragmenting its habitat, into, say, 10 small, completely isolated subpopulations.
Okay, 10 little islands of the species.
Now, drift starts acting independently in each of those 10 small populations. Each one starts its own random walk for every gene. Because the random walks are independent, population 1 might randomly lose allele A, while population 2 randomly loses allele B, they will inevitably start to drift apart genetically.
So even if they started identical, and even if their environments are identical, just because of random chance in each separate group, they'll end up looking genetically different after enough time.
Exactly. Population 1 might fix allele A, population 3 fixes allele B, population 7 fixes allele A, and so on, purely by chance. This divergence due to drift alone is a major reason why isolated populations often have distinct genetic profiles.
Okay, that makes sense. Now, the fifth feature sounds a bit more complex. Something about fixing alleles regardless of selection and a heterozygote deficit.
Right. Feature 5. Drift can fix alleles regardless of selection, and it creates a heterozygote deficit in pooled populations.
Yeah.
Let's take the second part first, the heterozygote thing, because it trips people up.
Yeah, the source said if you look at all those diverging populations together as one big group, you find fewer heterozygotes than you'd expect, even if within each small population things look fine, according to Hardy-Weinberg. How did that work?
Okay, let's use our example of 10 small populations. They all start with 50% big A and 50% little a alleles. So lots of heterozygotes, A individuals initially. Now let drift run for many generations. As we said, they diverge. Population 1 randomly fixes big A. Everyone becomes AA. Population 2 randomly fixes little a. Everyone becomes A. Population 3 fixes A. Population 4 fixes A, and so on. Maybe half fix A and half fix A, just by chance.
Okay, so within population 1, everyone's AA. Within population 2, everyone's A. No heterozygotes left in either one.
Right. Now imagine you, the scientist, come along and you don't know they're separate. You just take samples from all 10 populations and pull them together into one big data set. What do you see in your pooled sample? You see lots of AA individuals from the affixed populations and lots of exiguals from the affixed populations, but almost no AA heterozygotes because drift eliminated them within each individual subpopulation by driving frequencies to 0 or 1.
Ah, so the pooling creates the illusion of a heterozygote deficit. The variation isn't gone overall, it's just been sorted between the populations. You've separated the A's and the A's into different groups.
Exactly. The loss of heterozygotes happened within each small group, and when you mix those groups back together statistically, you see the deficit, and the first part of Feature 5. Because drift can be so overwhelmingly strong in small populations, it can actually drive an allele to fixation, even if that allele is slightly deleterious. If selection's pole S is weaker than drift's random push, 1 in A, chance wins, even if it's bad for the organism in the long run.
Okay, that forward-in-time view of drift, the random walk, is making sense. But you mentioned modern genetics often looks backward in time, this idea of the genealogy of genes. How does that work?
Right, thinking backward gives us a different, really powerful perspective. Instead of tracking frequencies forward, we take all the current copies of a specific gene in a population today, and we trace their ancestry backward through time. It's like building a family tree, but just for that one gene. You follow your copy back to your parents' copy, then their parents' copy, and so on. You do this for everyone's copies.
That sounds incredibly complicated for a big population. What's the pattern we see when we trace these gene lineages backward?
The key process you observe going backward is coalescence. As you trace lineages back, they start merging. 2 gene copies today might trace back to the same single gene copy in an ancestor 5 generations ago. That merge point is their coalescence point.
And that ancestor holds their most recent common ancestor, the MRCA. But just for that specific gene, right? Not the whole organism's ancestor.
Absolutely crucial distinction. Yes, the MRCA for that one gene. Different genes in your genome will have different MRCAs tracing back to different ancestors at different times. Your mitochondrial DNA might coalesce relatively recently, while some nuclear gene might coalesce much, much deeper in the past. Each gene has its own unique family tree, its gene tree. And as we trace these lineages backward, what happens? Some lineages just die out.
Right.
If an individual fails to pass on their copy of the gene, that line of descent vanishes when looking backward. So the number of distinct gene lineages can only decrease as we go further back in time.
They merge and they die out.
Until eventually, all the lineages currently present must inevitably coalesce back to a single ancestral gene, copy of the ultimate MRCA for all the current copies of that gene in the population. That coalescence is the historical footprint of genetic drift.
Okay, so the shape of that gene tree, how quickly the lines merge back, how deep or shallow it is, must tell us something about the history, like how strong drift was.
Exactly. If lineages coalesce really quickly, making a shallow bushy tree, it means drift was very strong. If they take a long, long time to coalesce, making a deep, spindly tree, it means drift was weak. And to put a number on that strength, to really quantify how powerful that random wind of drift has been for a population, we use the effective population size, usually written as a nuller.
No, okay, this sounds important, and you emphasize it's not the same as the census size and the actual head count. Why the difference? If I count 10,000 fish, why isn't none just $10,000?
Right. Numbers is a theoretical concept, a calculated value. It's defined as the size of an idealized population, one that perfectly fits assumptions like equal sex ratio, random mating, constant size over time, that would experience the same amount of genetic drift as the real-world population we're actually studying.
So it's translating the messy reality into a standardized drift strength measure.
Pretty much. And $none is almost always smaller, sometimes much smaller, than the census size N, because real populations are messy. They violate those ideal conditions. For example, if you have a harem system with 1 male breeding with 99 females, your census size N is 100, but nearly all the genes getting passed to the next generation go through that 1 male. The effective size, reflecting the bottleneck through the male lineage, will be closer to 4 than 100. Unequal sex ratios drastically reduce no allelers.
What else lowers malls?
Fluctuations in population size over time are huge. If a population booms and busts, the nullers is much more strongly influenced by the bust periods, the low points. Remember the elephant seals. Their long-term dollars is dragged down by that bottleneck of just 20, 30 individuals, even though there are 170,000 now. Nullers is mathematically calculated using the harmonic mean of the size across generations, which is heavily weighted by the smallest values.
Okay, so Nullers gives us the true measure of drift's power, and you said it connects to the timing of coalescence?
It does, directly. For a typical gene evolving neutrally in a diploid population, the average time you have to go back to find the MRCA of any two randomly chosen gene copies is 2 genome generations.
2 times the effective population size in generations.
Yep. So Gen A sets the timescale for genetic history within a population. And this reveals amazing contrasts. Take the fruit fly, Drosophila melanogaster. They exist in enormous numbers. Their estimated genus is huge, maybe around 1 million. So 2 densities is $2 million generations. That means, on average, any 2 copies of a typical fruit fly gene trace back to a common ancestor living 2 million generations ago. That's incredibly deep genetic history, reflecting very weak drift.
Wow. Okay, compare that to something with a smaller dollar.
It's chimpanzees.
Yeah.
Much lower population numbers historically. Their estimated dollars is maybe only around 20,000, so 2 densities is 40,000 generations. The average coalescence time for chimp genes is much, much shallower. Their gene pool has been shaped by much stronger drift, mixing things up more rapidly over time compared to the flies. It shows no $1, not just N, dictates the tempo of drift.
So Nostr captures the long-term average drift. But there are also these moments of really intense sudden drift. We talked about the population bottleneck, the temporary crash, like the elephant. Locked variation stays lost.
Exactly. And the other big one is the founder event. This happens when a new population is started by just a few individuals migrating from a larger source population. Those founders carry only a small, random sample of the genetic diversity from the original population. So the new population starts off with reduced variation, and because it's small, immediately experiences very strong genetic drift.
And we don't need to look at birds on islands for this, right?
No, yeah.
The book uses human history or expansion out of Africa as a prime example of founder events.
It's a perfect case study. Modern humans originated in Africa, which had the largest, most diverse populations. As small groups migrated out of Africa, then out of the Middle East, then into Europe, Asia, and eventually the Americas, each step involved a small number of founders carrying only a subset of the variation from where they came from.
Like a series of bottlenecks, one after another, as people spread across the globe.
Exactly, a serial founder effect.
Yeah.
And the genetic evidence is crystal clear. If you measure genetic diversity, say heterozygosity, in human populations worldwide.
It's highest in Africa.
Highest in Africa. And then it steadily decreases the farther away a population is from Africa, following the migration routes. Less variation in the Middle East, even less in Europe and Asia, less still in Native American populations. Each founder event randomly filtered out some variation. It's a beautiful, large-scale demonstration of drift-shaping genetic patterns through migration history.
That's incredible. It maps our journey just by looking at how much variation was lost by chance along the way.
And it really hammers home that point about fluctuating populations and alleles. Remember, now a dollar tends to reflect the minimum size a population experiences.
Right. You mentioned the influenza virus having a tiny dollar to sell despite massive outbreaks. How does that work again?
Think about the flu's life cycle. Billions might get infected in a bad year. Census size, N, is enormous during the peak. But how does the next season's epidemic start? It doesn't start from billions of viruses. It starts from a tiny number of viral lineages that successfully survive between seasons, maybe in the other hemisphere, or low-level circulation, and then successfully transmit to new hosts to kick things off again. Each transmission event, each jump between hosts, each survival over the summer, these are all potential bottlenecks. So the effective number of viral particles that actually perpetuate the lineage from one outbreak to the next is very small, maybe only a few hundred.
So that tiny transmission bottleneck dictates the nullarbacks, meaning the virus, despite its vast numbers at times, is highly susceptible to random genetic drift between seasons.
Precisely. Chance plays a huge role in which flu strains survive and seed the next wave. Okay, so we've firmly established drift is a major force, especially in small dollar populations, and its main effect is removing variation. Now let's connect that back to how much variation we actually expect to see in a population.
Right. We measure that variation using heterozygosity, symbolized as pi. And there's a theoretical link between drift and the rate at which new variation arises.
Yep, the neutral theory gives us a really neat baseline expectation. For mutations that are selectively neutral, they don't affect fitness, the expected level of heterozygosity in a diploid population at equilibrium should be approximately approx 4 Na.
Okay, let's break that down. 4 times the effective population size times the neutral mutation rate.
Right. Definitely is the rate at which new neutral mutations pop up per gene per generation. Devil reflects how quickly drift removes that variation. So the amount of variation you see is a balance between the input mutation and the loss, drift.
So this formula predicts big delta means weak drift, so variation sticks around longer, leading to high ladles like the fruit flies.
Exactly. And small delta means strong drift, variation gets lost quickly, leading to low plays like the post-bottleneck elephant seals. It gives us a theoretical yardstick.
And we can use that yardstick to understand why variation isn't spread evenly across the genome, right? Some parts are way more variable than others.
Absolutely. One of the most striking patterns is that non-coding regions of the genome, like introns, the bits snipped out of genes, or the DNA between genes, tend to have much higher heterozygosity than coding regions, exons, the parts that actually code for proteins.
Why would that be? What's special about coding regions?
It comes down to selective constraint and a specific type of natural selection called purifying selection. Think about it. A random mutation hitting a non-coding region often doesn't do anything functionally. It's likely neutral. So it's governed mainly by mutation introducing it and drift, potentially fixing or losing it. It contributes fully to that $40 expectation.
It's free to vary.
But a random mutation hitting a coding region, an exon, that's much more likely to change the amino acid sequence of the protein. And changing a protein is often bad news. It might mess up its structure or function. So most non-synonymous mutations, those that change the amino acid, are actually deleterious, maybe slightly, maybe severely, and natural selection jumps in.
Purifying selection, it cleans out the bad stuff.
Exactly. Purifying selection efficiently removes these harmful mutations from the population, often before they can drift up to any noticeable frequency. Because they're constantly being purged, they don't contribute much to the standing variation we observe in exons.
So coding regions are under tighter quality control by selection, keeping variation low, while non-coding regions are freer to accumulate neutral changes via drift.
Precisely. And this constraint is so fine-grained, you can even see it within a single codon, those 3-letter DNA words. Remember, the genetic code is redundant. Changes to the third position of many codons are often synonymous. They don't actually change the amino acid specified. So these third position mutations are often effectively neutral.
Ah, so they behave more like non-coding DNA, free to drift?
Largely, yes. But changes to the first or second position of a codon almost always do change the amino acid. They're non-synonymous, and therefore they're much more likely to be deleterious and get zapped by purifying selection.
So if you sequence a gene like, say, the alcohol dehydrogenase gene in fruit flies, you'd expect most of the variation within the exons to be at those third positions.
That's exactly what you see. The textbook mentions a study of AD, where out of 17 different polymorphisms found within the exons, only one was non-synonymous. All the rest are synonymous changes, mostly at third positions.
Wow, that's purifying selection caught in the act, basically preserving the protein sequence while letting the neutral parts wiggle around due to drift.
It's a beautiful illustration of constraint. Which brings us to the main event, the tug-of-war between drift and selection. When do these forces really clash?
Yeah, if you have an allele that's, say, slightly beneficial, selection wants to increase it, drift is randomly pushing it up and down, who wins?
It all boils down to comparing the strength of selection versus the strength of drift. We measure selection strength with the selection coefficient, how much fitter one allele makes an individual compared to another. A small s means a tiny advantage. And we measure drift strength by essentially somewhere. Remember, small L's means strong drift, so dollars is large. Large $, means weak drift, so one allele is small.
Okay, so it's S versus $.
And the rule of thumb is pretty straightforward.
Yeah.
For selection to reliably overpower drift and drive a beneficial allele to fixation, or purge a deleterious one, the selection coefficients need to be significantly larger than the strength of drift, a law or dollar.
So $1 means selection wins.
Selection wins. The allele's fate is determined by its fitness effect.
But what if it's the other way around? What if the alleles effect is really subtle, maybe only a tiny advantage, so Sellers is very small? And maybe the population isn't huge, so $dollars are relatively large. What if Seller does L?
Then drift wins. Even if the allele is slightly beneficial, its fitness effect is so swamped by the random noise of drift that selection basically can't see it. The allele is effectively neutral.
And its fate is just random chance. It might get fixed, might get lost, purely by luck.
Purely by luck. This difference in whether selection or drift dominates has massive implications for how precisely species can adapt, depending on their dollars.
Right. The book uses this analogy. It's like comparing a high-precision scientific balance scale to an old, rusty bathroom scale.
That's a perfect analogy. Think of species with enormous dollars, like our Drosophila friends, nanoprox or $1 million. For them, the strength of drift, $1 is incredibly tiny, like 10 whole. Their population genetics acts like that hypersensitive scientific balance. It can detect and respond to even minuscule fitness differences. Selection can effectively favor an allele, even if its advantage, S, is just, say, $10.
Because $10 is still much bigger than 10 to 6.
Exactly. And this allows for incredibly fine-tuned adaptations. The classic example is codon bias. In Drosophila, selection actually favors using certain synonymous codons over others, probably because they are translated slightly more efficiently or accurately. The fitness difference, S, is minuscule, but because $, is huge, selection can still push the population towards using the optimal codons.
Okay, now compare that to the rusty bathroom scale, a species with a small dollars.
Let's take gray whales.
Oh.
The number may be around $142. For them, the strength of drift, dollar to dollars, is $10. Now, that same tiny fitness advantage of $10 from the codon bias, it's completely invisible to the whale population. $10 is much smaller than $10.
So drift dominates.
Drift dominates. The whale population treats that potentially beneficial codon usage as effectively neutral. Its frequency will just bounce around randomly. It might get lost, even if slightly advantageous. So the punchline is, adaptation in small nihilist species is inherently less precise. They can only adapt to mutations with relatively large fitness effects. Subtle improvements get lost in the noise of drift.
And this brings us to a really worrying consequence of strong drift in small populations. The inbreeding load. This sounds bad.
It is bad. It's one of the major concerns in conservation biology. Remember Feature 5, where drift could fix alleles regardless of selection? The inbreeding load happens when drift becomes so strong, because the X is tiny, that it starts randomly fixing alleles that are actually slightly deleterious. Selection should be getting rid of them, but if their negative effect S is smaller than the power of drift, chance can override selection and fix the bad allele anyway.
And if this happens at lots of different genes,
Then the overall fitness of the population starts to decline. Individuals become less healthy, less fertile. That accumulated burden of slightly harmful mutations fixed by drift is the inbreeding load.
and we see this in real life, not just theory.
Oh, absolutely.
Huh?
Captive breeding programs constantly fight against it, and the book gives a fantastic, dramatic example from the wild. The Swedish adders, Viper aberus. There was this one population in Sweden that got totally isolated by farming and forestry. Its size crashed to fewer than 40 snakes. Tiny dollar, massive drift.
So they became extremely inbred. What happened to them biologically?
The consequences were severe. Researchers noticed really low reproductive success. Many females were infertile, and a high proportion of the offspring that were produced were stillborn or had birth defects. Classic signs of inbreeding depression, likely caused by deleterious alleles drifting to high frequency or fixation.
The population was basically spiraling towards extinction because its own genes were failing it.
Pretty much. It wasn't that the environment suddenly got worse, it was internal genetic decay driven by drift.
But then there was an intervention, right? A kind of genetic rescue.
Yes, it's a famous study. To combat the inbreeding load, researchers captured 20 male adders from a larger, healthier population nearby and introduced them into the isolated, struggling population. They let them breed for a few years.
So basically injecting fresh genetic variation, bringing back the healthy alleles that had likely been lost by drift in the small population.
Exactly. It was gene flow aimed specifically at counteracting drift and inbreeding, and the results were almost immediate and incredibly positive. Offspring survival rates shot up. The number of snakes born alive increased dramatically. The population size started to rebound strongly. It was clear proof that the original problem was the genetic load caused by drift in a small population, and that even a small amount of introduced variation could reverse the decline.
Okay, so drift is powerful, especially when net is small, and it generally removes variation or even fixes bad stuff. But then, in the late 60s, Motu Kimura came along and proposed something really radical, the neutral theory of molecular evolution. Wasn't his idea basically that most evolution at the DNA level is actually driven by drift, not selection?
Yeah, it was a bombshell. The prevailing view had been that nearly all changes fixed between species were adaptations driven by positive selection. Kimura flipped that on its head. He argued that the vast majority of mutations that actually get fixed in evolution, the differences we see when comparing DNA sequences between species, are selectively neutral. They have no significant effect on fitness.
Hmm.
And if they're neutral, what's the main force determining their fate?
It has to be genetic drift. If S was 0, then only drift matters.
Precisely. So Kimura proposed that most molecular evolution isn't adaptation. It's just the result of neutral mutations arising and then randomly drifting to fixation over long periods. Selection, he argued, was mainly acting in a purifying way, removing the deleterious mutations, while positive selection fixing advantageous mutations was actually quite rare at the molecular level.
That sounds like a huge claim. What made people take it seriously? Did it explain something existing models couldn't?
It did. Its greatest triumph was providing a simple, elegant explanation for an observation that had been puzzling biologists: the molecular clock.
Ah, the idea that genes seem to evolve or accumulate differences at a surprisingly constant rate over time. Like you can compare the same gene in humans and chimps versus humans and orangutans and estimate divergence times based on the number of differences.
Exactly. People observed this clock-like accumulation of changes, but it wasn't immediately clear why it should be so regular across different species with different generation times or population sizes. The neutral theory provided the mechanism. It showed mathematically that the rate at which neutral mutations become fixed in a population is equal simply to the rate at which neutral mutations arise.
Oh, wait, the rate of fixation is just the mutation rate? How does population size drop out? That seems counterintuitive.
It does seem weird at first, but think it through. In a diploid population of size Nero, there are 2 Nisegeli gene copies. New mutations arise at a rate of monad per gene copy per generation. So the total number of new mutations entering the population each generation is 2 Nisegeli times money. Now, if a mutation is truly neutral, what's its initial probability of eventually drifting all the way to fixation? It's just its initial frequency. Since it starts as a single copy out of 2 Nisegeli copies, its chance of eventual fixation by drift alone is $1.
Okay. Rate of new mutations is 2 to fixation is $2. Chance of fixation is $1.
So the overall rate at which new neutral mutations arise and eventually get fixed is the product of those two things. 2 in a days,
Yeah, the two cancels out.
the 2 in a d cancels out. The rate of neutral substitution fixation is just equal to the neutral mutation rate, NuDale. It's independent of population size. So if the neutral mutation rate is reasonably constant over time for a given gene, the neutral substitution should accumulate at a steady clock-like rate. That's the molecular clock explained by neutral drift.
And that divergence between two species is then just $ol biotol all 2, where it's a time since divergence.
Yep, the tuna comes in because changes accumulate independently on both lineages, leading back to the common ancestor. It provides a powerful way to estimate how long ago species split based on their genetic differences, assuming neutrality.
Okay, but how do we test this? How do we know if a gene is evolving neutrally? We need something where we're sure selection isn't interfering.
That's where pseudogenes come in. They are the perfect natural experiment. Pseudogenes are basically broken copies of real genes. They've accumulated mutations that stop them from being transcribed or translated. They're genetic fossils, non-functional.
So any mutation that happens in a pseudogene doesn't matter. It has no effect on the organism's fitness because the gene doesn't do anything anyway.
Exactly. Mutations in pseudogenes are about as close to perfectly neutral as you can get. Therefore, according to the neutral theory, they should be evolving, accumulating substitutions at the maximum possible rate, the neutral mutation rate, 40 in dollars.
And is that what we see? Are pseudogenes fast evolving?
They absolutely are. When you compare evolutionary rates across different parts of the genome, pseudogenes are consistently among the fastest evolving sequences. They accumulate differences rapidly, just as predicted if drift is the primary driver and selection isn't constraining them.
And conversely, bits under strong selection should evolve slowly.
Right. We can test the flip side using purifying selection. Look at non-synonymous sites in critical functional proteins. We know these are under strong constraint. Changes are often bad. And indeed, these non-synonymous sites accumulate substitutions much more slowly than synonymous sites, which are often nearly neutral, or pseudogenes. The slower rate reflects selection weeding out most changes.
So the relative rates make sense. Pseudogenes fastest, neutral. Synonymous sites intermediate, mostly neutral. Non-synonymous sites slowest. Strong purifying selection.
It paints a pretty coherent picture, supporting the idea that much of the background molecular change is indeed neutral drift.
So does that mean Kimura was totally right? Is positive adaptive selection rare at the molecular level?
Well, that's where the nuance comes in. The neutral theory was revolutionary. and provides an essential null hypothesis, what evolution looks like without selection. But modern research, with vast amounts of genomic data, suggests positive selection might be more common than Kimura initially thought. Some studies, especially comparing closely related species like different Drosophila or even humans and chimps, estimate that maybe 30%, 40%, even up to 50% of the amino acid differences fixed between them might actually be due to positive selection driving beneficial mutations, not just neutral drift.
So the reality is a mix, a lot of neutral drift forming the background, but also a significant amount of adaptive evolution layered on top.
That seems to be the current consensus. The molecular clock isn't perfectly constant either, rates can vary, and fluctuating selection can also sometimes mimic clock-like patterns. But the neutral theory remains incredibly important as the baseline model of evolution driven purely by mutation and chance.
Okay, so if the genome is this mix of neutral changes driven by drift and adaptive changes driven by selection, how do we actually tell them apart? If we see a difference between two species, how do we know if it was just random luck or if it was genuinely adaptation, selection choosing the better version? We need tools to find those signatures of selection.
Absolutely. Distinguishing selection from drift is the central goal in evolutionary genetics, and we have developed some pretty clever methods. One of the most fundamental is the DN-DSA ratio.
Okay, DN over DS. What do those stand for again?
So when you compare a protein coding gene between two species, you count two kinds of substitutions or fixed differences. D dollars is the rate of non-synonymous substitutions, those DNA changes that did alter the amino acid sequence. D dollars is the rate of synonymous substitutions, those DNA changes, often at the third codon position, that did not alter the amino acid sequence.
And the logic is that the synonymous ones are mostly neutral, so they reflect the background rate of change due to mutation and drift.
Exactly. The AD serves as our baseline, our neutral expectation. DAD, the non-synonymous changes, are the ones that selection can potentially see because they change the protein. So by comparing the rate of potentially functional changes to the neutral background rate, we can infer the dominant type of selection that's been acting on that gene.
How does the ratio tell us that? What are the key thresholds?
There are three main interpretations. If DAD is less than 1, it means non-synonymous changes are accumulating much slower than synonymous ones. This is the signature of purifying selection. Selection is actively removing deleterious amino acid changes, keeping the protein sequence conserved. This is the most common scenario for most genes.
Okay, that makes sense. What if the ratio is close to 1?
If DND is equal to 1, or very close to it, it suggests that non-synonymous changes are accumulating at roughly the same rate as neutral synonymous changes. This implies neutral evolution, where drift is the primary driver, and selection isn't strongly constraining or favoring amino acid changes. Perhaps the gene's function is relaxed.
And the exciting one, the signature of adaptation.
That's when DNDS is greater than 1. This means non-synonymous amino acid changing substitutions have accumulated faster than the neutral baseline rate. The only plausible way for that to happen is if positive selection has been actively favoring and rapidly fixing beneficial mutations that change the protein's function. A ratio significantly above 1 is strong evidence for adaptive evolution acting on that gene.
Do we see this often? Are there examples?
We do see it, often in genes involved in arms races, like immune system genes fighting pathogens or reproductive proteins involved in sperm competition. The textbook mentions the BRCA1 gene in humans. While it's known for cancer susceptibility links, when compared across primates, parts of the gene show a DNDS ratio greater than 1. This suggests that during primate evolution, positive selection drove rapid changes in this gene, perhaps related to DNA repair or other functions, not just cancer.
Interesting. So DNDSSI looks at fixed differences between species. Is there a way to detect selection using variation within a species too?
Yes, and that leads to more sophisticated methods like the McDonald-Kreitman test or NK test. This test is really powerful because it compares divergence between species with polymorphism within a species, using synonymous sites as the neutral benchmark throughout.
How does combining within species variation, polymorphism, and between species difference, divergence help?
The core idea is that under pure neutrality, just mutation and drift, the ratio of non-synonymous to synonymous changes should be the same for polymorphisms within a species as it is for fixed differences between species.
Okay, the ratio of an N mass of variation should equal the ratio of N to solid divergence if everything's neutral.
Right. But if positive selection has recently driven beneficial non-synonymous mutations to fixation, it changes that balance. Those beneficial mutations contribute a lot to non-synonymous divergence. They become fixed differences quickly, but they contribute very little to non-synonymous polymorphism because they sweep through the population fast, not lingering as variation.
Ah, so positive selection inflates the meta-Noller ratio for divergence compared to the non-Noller ratio for polymorphism.
Exactly. The MK test compares these ratios in a statistical framework. If it finds significantly more non-synonymous divergence than expected, based on the polymorphism levels, using synonymous sites to set the neutral expectation, that excess divergence is attributed to adaptive evolution.
That sounds like a much more rigorous way to detect adaptation. Has it been used much, say, in humans?
Extensively. MK tests or related methods have been crucial for identifying genes that likely underwent positive selection during human evolution. The book notes that genes involved in things like transcription factors controlling other genes, hormone receptors signaling, and immune responses often show strong MK test signals. This suggests these complex systems were key targets of adaptation as humans evolved.
Okay, so we can hunt for selection gene by gene, but can we look across the whole genome? Maybe to find genes responsible for adaptation to a specific local environment?
Yes, that's the idea behind genome scans for selection. Here, instead of focusing on one gene, you compare genetic variation, usually allele frequencies at thousands or millions of SNPs, single nucleotide polymorphisms, across the entire genome between two populations.
And you're looking for outliers, places where the populations look way more different than expected just by drift.
Precisely. Imagine you have two populations that split relatively recently and now live in different environments. Because of their shared history, most of their genomes should still look pretty similar. Allele frequencies at most SNPs should be highly correlated due to shared ancestry and neutral drift acting similarly in both. But if one population has adapted to its unique local environment, the specific genes involved in that adaptation should show unusually large frequency differences between the two populations. Selection has pushed the beneficial alleles up in one place and maybe down in the other. So you scan the genome looking for SNPs or regions that are extreme outliers in terms of frequency differences. Fs is a common statistic used here. These outliers are strong candidates for being under local adaptation, driven by selection.
And the classic example here is the adaptation of Tibetans to high altitude.
It's a perfect case study. Tibetans have lived successfully at extremely high altitudes with low oxygen levels for thousands of years. Han Chinese are their closest relatives genetically, but live primarily at lower altitudes. So researchers did a genome scan comparing SNP frequencies between Tibetan and Han Chinese populations. Just as expected, for the vast majority of SNPs across the genome, the frequencies were very similar, reflecting their recent common ancestry.
But there were outliers.
There were striking outliers. A couple of SNPs in particular showed enormous frequency differences. These were located within or near a gene called EPAS1.
And what does EPA1 do?
It's a key gene involved in how the body responds to low oxygen, hypoxia, and the frequency difference for the beneficial allele was massive, something like 90% frequency in Tibetans versus only 10% in Han Chinese.
A 90 versus 10 split. That's huge. Is that difference too big to just be random drift since they separated?
Way too big. Given how recently the populations diverged, achieving an 80% frequency difference at a single gene through random drift alone is statistically almost impossible. It's an incredibly strong signature of very recent, very powerful positive selection acting on EPAS1 in the Tibetan population, favoring variants that help them thrive in the thin air of the high plateau. It beautifully demonstrates how genome scans can pinpoint the specific gene's underlying local adaptation, cutting right through the background noise of drift. #TagYocheng So wrapping things up for today, I think the big takeaway is that evolution isn't just about the best adapted surviving. Random genetic drift is this incredibly powerful, ever-present force. It's especially potent when populations are small or go through bottlenecks, and it's the main reason genetic variation gets lost over time within a population.
Yeah, and we saw the consequences it can lead to divergence between populations just by chance, and maybe more worryingly, it can cause slightly harmful mutations to become fixed, leading to that inbreeding load, like we saw with the Swedish adders. That was quite the story.
Definitely. But we also saw that even though the neutral theory highlights the importance of drift, especially at the molecular level with the clock, we now have these really sophisticated tools, the DNA-to-ozone ratio, the MK test, genome scans that let us find the specific fingerprints of positive selection, distinguishing adaptive changes, like in BRCA1 or the Tibetan's EPAS1 gene, from the background noise of random chance.
Right. So we've now looked really closely at the two main engines driving evolution within populations. Selection giving direction, and drift adding that element of chance. But, populations don't live in perfectly mixed isolation. There's geography, there's distance, individuals move around.
Exactly. What happens when we add space to the equation? How does the movement of individuals between populations, what we call gene flow, interact with drift and selection? Does it counteract the divergence caused by drift? Can it spread adaptations?
That sounds like where we need to go next, exploring evolution across landscapes, considering how geography and migration shape the patterns of genetic variation and adaptation we see in the real world. So definitely tune in for that. Thanks for diving deep with us today on Bookfulness. Join us for the next deep dive.